So, hello and welcome, and thank you for joining us. My name is Vishal Dikshit. I am a senior NGS field application scientist at Twist Bioscience. And next 15 minutes, I would like to show the new technologies what Twist Bioscience is working on to make clinical and diagnostic lab to generate best-in-class sequencing data. So, little background about Twist Bioscience as a company. So, we founded in 2013. Our headquarters is in South San Francisco, and we have multiple offices in different parts of the world. And recently, we have launched a factory of the future in Portland, Oregon. So, what makes Twist different from everyone else? So, as you can see, the linchpin of Twist Bioscience business is Twist silicon platform, which is based on microfluidics and the nanowells. So here you can see, So here you can see the oligos, which are synthesized in this Twist silicon platform. So how we are different from the other vendors. So if you see the standard workflow of synthesizing the oligos is based on standard labware, which is either 96 well plate. So the disadvantage is that you need one well for each gene. However, when it comes to Twist, using microfluidics and the nanowell on silicon platform, we can create 1 million oligos per chip. So at the end, one wafer, we are creating 9,600 genes, which is very useful for the customer who are really interested in custom panels with short turnaround time. So what's next? We have synthesized the oligos, but these oligos are not enough to perform an NGS assay. So we have to amplify these synthesized oligos to have enough mass for the experiment. So what we do? So if you see the uniform, there's a first graph showing the uniform synthesis of the oligos in the standard 96 well plate or our Twist silicon platform. However, we have to amplify these probes to have a greater or the mass for the NGS experiment. So if you see the standard amplification method, what other labs or other vendors are using, it brings biased in probe representation. And that you see when you see or upload your data on IGV plot. You will see some hills down and some very high hills. Hence, in a nutshell, it's a biased in probe representation, which is making hybridization not efficient. However, in Twist, we use proprietary amplification method, where once the oligos are synthesized, we use amplification, again, proprietary amplification method to have highly uniform probe pool, which you can see on an IGV plot, the really good uniform coverage for all the targeted regions. And also, what we do, we have a Twist double-stranded DNA probes, which means these double-stranded probes, when you do the hybridization, it hybridizes both strands of the DNA. So which means in single assay, you can have a double information about that region. So this is, again, very useful if you're working for detection of ultra-low VAF in case of oncology. So utilizing all these proprietary technologies, we launched first of our human core exome in 2018 at AGBT. But if you see, we are continuously working for the welfare of community. As you can see, in 2020, we launched SARS-CoV-2 panel along with the synthetic controls, which really helps the communities to overcome the pandemic. And also, we are not only working for DNA, we are also working for the targeted methylation. We launched a few panels for the targeted methylation or epigenetic studies. And jumping to 2024, which I will show the new innovation products from the Twist Bioscience. So I will be showing you cell-free DNA library prep kit. I will be talking about RNA-seq portfolio, and finally, our first IVDR exome. So let's go to our products. Yeah, so I would like to introduce our new, recently launched Twist Precision DX, or CE-IVD workflow for the Twist exome, which involves complete workflow. So if you can see, the green part represents the Twist kit. So it's all CE-IVD mark, which is Twist Precision Prep, which is based on enzymatic fragmentation. And then we also have enrichment DX kit, along with Twist Precision Exome DX panel. So this contains not only exome genes, but also we have spiked in the mitochondrial region. So once you have a CE-IVD exome from the Twist, you have a complete coverage of genome. So as you know, like Twist is quite agnostic. So what we did for other labs who are not interested in exome, but the small custom panels, like oncology lab, pathology lab. So what we are also panel agnostic, which means you can still use our IVD marked Twist Precision library prep and enrichment, and Twist can help you to benchmark your custom panel as a CE-IVD mark. And then we also have like twist precise exome DX panel that you can use it with any integrative other workflow. And finally, the third product is a complete product, which means we provide you library prep kit, standard hybridization, and the exome as a CIVD marked. So now, the DNA is very popular, but personally, I really like to explore the RNA. So the Twist recently also launched Twist RNA sequencing solutions. So we are providing wide array of RNA solutions. For example, if you see, we have a whole transcriptomics RNA sequencing, which you can use for unbiased transcript quantifications. Our bioinformatics team also worked to design RNA exome, which is very different from DNA exome. We really focused on the coding content of the genome and designed this Twist RNA exome, which is very important or very useful if you have like small sequencer and you need to have more data with the fewer reads. And also, these applications can be variety, like fusion detection, novel fusion detections. And finally, as you know, we are expert in designing custom panels. So we are also having focused, targeted RNA-seq. So the best thing is not we can always produce for short read, but we can also do long read, like full-length iso-seq enrichment with the long read custom panel. And also, it's already being used by some of the oncologists for the fusion detections. So this is one of the really nice result I would like to show you, because right now, when we talk about RNA, we also talk about cell-free RNA, what is the amount of RNA that we can use to convert into the library. So if you see the first graph, in the y-axis is the mass input, and the y-axis is the gene detected. And green represents the good quality or a universal reference RNA, and the orange represents the FFP sample. So you can appreciate even with the 1 nanogram or the 10 nanogram of RNA, the sensitivity of assay is still not sacrificed. You can still detect more than 15,000 genes even with 10 nanogram of RNA, FFP RNA. So this was one experiment that we want to replicate to see how robust this assay is. So here you can see some replicates. So the first plot is 1 nanogram, 10 nanogram, and 100 nanogram, and you can really see the highly correlation between two interreplication experiments. So again, Twist library prep can be used for really ultra-low amount of RNA, and it's independent of any quality of RNA, for example, FFP RNA or universal good quality RNA. So now I would like to focus, like in last decade, there has been a lot of research going on liquid biopsies, for oncology, for NIPT. So this is general image. It shows that as a Twist Bioscience, we can provide you end-to-end workflow for liquid biopsy research. So we have Twist target enrichment system based on really ultra-low input of cell-free DNA. We have Twist UMI adapter system, which can be used for detecting ultra-low VAF. We have cell-free DNA standards. We have MRD panel for minimal residual diseases. And what I am going to talk today is a cell-free DNA library prep kit, which is, again, quietly appreciated by a lot of labs in Europe. So the key selling point of our library prep kit is reliable and robust performance. You can see, you can start with even 1 nanogram of cell-free DNA. And that's not, we are talking from my marketing team, but I have seen in the labs also that they are using 1 nanogram of cell-free DNA to convert library with high complexity. So generally, from 1 to 20 nanogram, you can really have a good library, cell-free DNA library. We have our in-house engineered T4 ligase, which brings greater library complexity or library conversion rate. And this all innovation-driven improved performance can be really helpful for oncology field and also for NIPTs. So this, again, if you see this bar graph, so I don't know if this is working, but if you, sorry. So if you see this bar graph, we utilize 15 nanogram of cell-free DNA with different VAFs, 0% VAF, and then the 0.25 VAF. And you can see with the Twist new cell-free DNA kit with the engineered T4 ligase, you have 2x improved performance with the conversion efficiency. And this is very important if you are working with oncology cell-free DNA, where conversion rate is very important. And then we also looked into the library complexity with using the UMI filter 100 and 2111. And you can see even with high stringency, which is 211, you can see very high mean target coverage compared to other vendors. So again, it's really quite cool instrument or a platform or product if you want to work on oncology, if you are working with oncology field. So here comes, like, today's now in the at ESHG conference, there are a lot of sequencers. So are we agnostic with all the sequencers, or we are just with Illumina standard workflows? So if you can see, all the sequencer roads comes to end at the Twist Bioscience, which means you can have any sequencer, but you can always utilize Twist Bioscience library prep or the enrichment for the enrichment. For example, with the PacBio, we're doing successfully long-read enrichments, and our 2 guest talks are for the long-read from the PacBio, and also we did some long-read IsoSec for the full-length RNA. And we also have a recent collaboration with Element Bioscience, so you can use our workflow on Element without doing the conversion. And also we recently launched beta program with Nanopore. So yeah, all sequencers perform powered by Twist. So you just can come to our booth if you have any sequencer, and we will provide you a solution. So this is just one example. So recently in London Calling, we launched together with Oxford Nanopore a beta program where you can use our Twist Alliance long-read PGX panel on ProMethane on GridION. And what we do, we also provide a demultiplexing script from the ONT. So again, this is the beta program for PGX panel, but it's not end here. You can design any long-read panel, for example, HLA panel, SMN1, SMN2 panel for long-read, and it can be worked very effectively on Oxford Nanopore. So again, if you have any question regarding long-read, you can visit our booth. So if you find anything interesting in my talk, so you can scan this barcode and we will contact you. So apart from this, it's just the latest innovation, but we are also working on ancient DNA. We are working on multiple NGS assays, so just feel free to contact us if you have any genomic experiment to perform. So we have 2 guest speakers for our corporate satellite symposium. So I would like to invite first of the speaker, Dr. Suzanne Kluit, if I spell right. So she's head of the Leiden Genome Technology Center in Leiden University Medical Center in the Netherlands. And she will be talking about using targeted long-read sequencing of pharmacogenes to improve drug response prediction. I hope I spelled right.
So thank you very much for the introduction, and thanks to Twist for inviting me to speak today. By the way, there's still some seats down here in the front. For those of you in the back, don't be shy. Come on down. So I'd like to begin today by giving you all a very brief introduction to the field of pharmacogenomics. So pharmacogenomics, or PGX for short, is what the name suggests. So it's the study of how each individual's genotype affects their drug metabolic phenotype. Now, current pharmacogenomics practice classifies individuals on the basis of this drug metabolism phenotype. So for any given drug, the majority of individuals in the population will be a normal metabolizer of this drug. However, there will be individuals in the population who actually break down this drug very rapidly. So they're what's known as ultra-rapid metabolizers, and this means they need a lower dosage of the medication for the drug to have its desired effect. Similarly, there will be individuals in the population who are poor metabolizers of a particular drug, and they need to have a much higher dosage of the medication for the desired result. And there may also be individuals in the population who have a contraindication for a particular medication, and that means they should be offered an alternative drug. Now, pharmacogenomics is actually really important because it's highly likely that everyone in this room contains at least one actionable pharmacogenetic variant in their genome. And that was nicely illustrated by a recent population study in Denmark, where they looked at 77,000 individuals and found that 99.9% of them carried at least one actionable variant, with the majority of people in the population possessing 3 or more actionable variants. So the consequence of this is that up to 10% of patients may suffer from an adverse drug reaction when starting a new medication. Now, in mild cases, this means they may have some nausea or fever, but in very severe cases, this can actually result in death. Now, luckily, with current pharmacogenomic practice, we're able to prevent up to 30% of these adverse drug reactions. But you can see we still have a long ways to go in the field until we bring this to 100%. Now, there's also a large cost to society, and it's estimated that billions of euros are wasted per year with unsuitable treatments, with individuals who need to come back to the clinic for dosage adjustments or for medications that need to be thrown away because they're unsuitable. So the current struggle in the field of pharmacogenomics is that these genes, they're really difficult to sequence. So that was nicely illustrated in a recent review article looking at common pharmacogenes on the x-axis. And what the bar graph shows you is the fraction of that gene that's located in a complex region of the genome. And by complex, we mean contained within a segmental duplication, a repeat, or maybe even a combination of the two. So you can see that for several of these genes, 100% of the gene is located in a difficult to sequence region. So these are very hard to see with short-read sequencing. So what we wondered at the LUMC a while ago was if long reads could actually help us resolve these difficult-to-sequence pharmacogenes. So my team partnered with Jesse Sven's lab in our Department of Pharmacy to see if we could now resolve these difficult-to-sequence genes with PacBio hi-fi sequencing. So we were very happy to have early access to one of PacBio's first hi-fi whole genome sequences on the HD002 individual from the Ashkenazi trio. And what Maaike van der Lee, a PhD student at the time, did is look at these common pharmacogenes and figure out, can they be phased? So similar to the previous slide, you see all of the genes with the fraction of the gene that's located in a complex region shown in the bar graph. But now we've added these circles. And the circles tell you whether or not that gene could now be phased. So can we resolve the alleles that the variants belong to? And we were really happy to see that for the majority of genes, more than 90% of the variants could now be resolved into their component alleles. So this might offer a lot of promise for us in improving the clinical workflow. So the first time we decided to apply long reads was to perform targeted amplification of the CYP2D6 gene. Now, CYP2D6 is a cytochrome P450 enzyme, and it's responsible for metabolizing an estimated 40% of commonly prescribed drugs, and it's also notoriously difficult to sequence with short-read sequencing. So what we did is use an amplicon-based workflow to identify full-length CYP2D6, as well as CYP2D6 deletions, duplications, and fusions with the nearby pseudogene CYP2D7. And we applied this amplification strategy on a cohort of women who were undergoing tamoxifen treatment for breast cancer. So we have very nice metabolic ratios of the metabolism of tamoxifen in the patients, and now we were able to also generate genotyping information from these women, with 561 moving forward into the second part of our study. So what Jesse Sven's team did is they first looked at the metabolic ratios according to the current clinical guidelines, where we've classified each individual as a poor, intermediate, normal, or ultra-rapid metabolizer on the basis of their genotype. We then looked at the metabolic ratio, which is plotted on the y-axis. And what I hope you can appreciate is that there's really a big spread in the data. So if we take a look at the normal metabolizers, shown here in the light blue. Oh, switch to this. So here you can see that some of the normal metabolizers actually break down the drug as poorly as our poor metabolizers over here. And similarly, we have many normal metabolizers who actually break down the drug as efficiently as the ultra-rapid metabolizers. So using the current clinical guidelines, we're only able to explain about 54% of the variation in our data set. But what Jesse's team did is they actually used each individual's genotype and phenotype information as input into a neural network. So we used machine learning to try to develop a better model to predict each individual's drug response. So if we do that, and we now plot the data on a continuous scale instead of a categorical scale, we now much more accurately can predict each individual's response on the basis of their genotype, and we're explaining about 79% of the variance in our data set. So of course, we were quite happy with these results, but we're only looking at one pharmacogene. So for our next study, we wanted to go a bit bigger. So for this, we decided to partner with Twist Bioscience to develop a method to perform long-read hybridization capture. And for this, we work with a custom panel targeting 21 of these pharmacogenomic loci. And we selected these loci on the basis of the Dutch Pharmacogenomic Working Group guidelines. We also included several HLA class 2 genes. And for us, we're really coming from a research background, so we wanted to look at the full gene body of these loci, so all of the exons and introns. And we also look 10 kilobases upstream of the transcriptional start site and 10 kilobases downstream of the transcriptional end site. Now, I hope there's some fellow methods geeks out in the audience, so I wanted to briefly walk you through the workflow that we set up. So we start by shearing genomic DNA to about 7 kilobases, and for this we use either Covaris G-tubes or diagenode megaruptor. We then proceed with a standard end-repair A-tailing, followed by adapter ligation. And then what we do is we perform pre-capture PCR, where we use an enzyme that's specific for long-range PCR. So we've been working with Takara LA-TAC for this. Twist recommends Millipore's KOD polymerase. After that, we pool the libraries, and we work with an 8-plex hybridization reaction. We then use probes that were designed by Twist, followed by Twist reagents for capture and wash. And finally, we perform a post-capture PCR, again using the same long-range polymerase. And then we move to PacBio's workflow, where we perform a standard Smart Bell prep. And the data I'm going to show you today was sequenced on our Sequel IIe platform. but you can also use PacBio's new platform, the Revio, to do this at a higher throughput and for a lower cost. So we began by benchmarking the performance of our panel, and for this we chose to work with samples from the Genome in a Bottle Consortium, because there's been a lot of efforts out there in the community to make really great truth sets for these individuals. And in particular, we worked with the Ashkenazi trio, so HG002 I mentioned earlier, as well as the parents HG003 and 4. So we first checked the performance of our panel, and we were really happy to see a very high on-target rate of 70%, with a good level of enrichment and an average target coverage of 118-fold for the genes in our panel. We also checked the variant calling in our samples versus the truth set, and we identified for SNPs, we have a precision of 99.3% and a recall of 97.5%. And keeping in mind that these are some very difficult to sequence genes, we are actually really happy with the results. For indels, we struggle a bit more, but this is still far and away much better than what we ever saw with the short-read sequencing. So I wanted to give you an impression of what the data look like. So here I've included an IGV screenshot of the CYP2B6 locus. So this is a 47 kb long gene, and we've also included the up and downstream regions, which you can see in the coverage plot here. And you can see that we can very nicely phase almost all of the PacBio reads into one of two haplotypes. And if we zoom in on one particular variant that's here in the 5' most exon, you can see really clearly that the child has a C to T SNV in one allele and reference in the other. And we can see that the child inherited the SNV from dad and the reference allele from mom. But even without having the trio information, we could still really clearly phase this individual into the two alleles. So now that we know our panel's working really well, we wanted to move to clinical samples. And for this, we're working with samples from the Allegro study coming from 3 sites in the Netherlands. And these are individuals who have received kidney transplants, and they've undergone long-term monitoring of their immunosuppressant metabolism. So we have really great drug metabolism longitudinal data for these individuals. And our goal with this study, similar to what we did with CYP2D6, is to eventually link each individual's genotype to their drug metabolic phenotype, with the goal of improving drug-gene interaction predictions. And for this study, in the end, we have nearly 200 patients who have finished the study, and we have access to their DNA. So what I'll show in the last part of my talk is the initial work that we've done characterizing the variants that we identify in this study. So we have 44 patients sequenced so far, whose results I'm going to present. And here we're just looking at 11 of the 21 genes that we have in the panel, and I'm only going to show the results from the SNVs. So what we found across our panel so far is nearly 4,900 variants in these 11 genes. And coming from the core region, so just the exons and introns, we see 3,200 variants. And per gene, we're finding several hundred variants across these 44 samples. So there's really a lot of unappreciated variation going on in the population. If we look at what's currently used in clinical practice, that's on the right, the star alleles, where variants are turned into an allele that's then used in the clinic. And we find that only 3% of the variants that we found are actually currently used in clinical practice. Now, to be fair, we are looking very broadly at the genes in this panel. So based on the variant effect prediction annotation, the majority of the variants that we identified are located intronic or up and downstream of the transcription start or end site, respectively. However, we also identified many potentially impactful variants based on the VEP classification. So we see 92 missense mutations, 34 splice site mutations, 6 frame shift, and 2 stop gain mutations. And we also wanted to search for other variants that might be impactful. So for this, we decided to look at the CAD score of some of the SNVs using the tool developed by Jason Jury's group. And here we're plotting all of the variants and the star allele variants using the CAD score. So you can see on the left in the category of frame shift variant, these are very deleterious. So they have a very high CAD score, above 15. So we decided to use a cutoff of 10 to try to identify other potentially impactful variants in our data set. And when we apply that threshold of 10, we find that 116 of the variants are potentially impactful. And this is on top of the variants we already identified with the VEP tool. So in total, we've now found 250 variants in our data set. That's 5% of the variants that we identified that may be potentially impactful. And of these, only 16% are currently used in clinical practice. So 84% of these potentially impactful variants are not used in clinical practice. So of course, this requires a lot of follow-up study and investigation, but I think it really shows the power of this approach to identify new variants. So I'd like to briefly summarize. So I showed you today that long reads can be useful to resolve segmental duplications and repeats within medically relevant loci. That if we use these phased pharmacogenomic alleles, and in this case CYP2D6, we combine that with metabolic data, we can improve drug response predictions. We developed a hybridization capture workflow together with Twist and PacBio that can be used to cost-effectively sequence and phase a large panel of pharmacogenomic loci. And if we sequence these dark regions of the genome, we can reveal many new potentially impactful SNVs. I'd like to conclude by acknowledging all the people that contributed to the unpublished work that I showed you today. On my team at the Leiden Genome Technology Center, that includes Luce and Rolf in the wet lab and Roberta for data analysis. I'd like to acknowledge our department chair who actually helped fund this project, Sylvia Vandermarl, and the team of Jeroen Leroux, specifically Redmar and Mark, who've been developing pipelines to help automate this analysis. And of course, this study wouldn't have been possible without the great collaboration with the group of Jesse Sven, in particular Maaike Vanderlee, who really pushed this project forward, and Chinyan and Lonica, who've done the variant analysis I showed on the last few slides. And I'd also like to acknowledge the great collaboration that we've had with Twist, in particular with Tina Hahn, and also the team at PacBio. And thank you all for joining and for your attention.
Thank you, Suzanne, for the nice talk. So now stage is open for Q&A, so feel free if you have any question to ask to Suzanne. Do you guys have any question for the Suzanne? So do you have any Q&A for Suzanne? Oh, we have.
Very nice presentation, thank you. I was wondering whether the panel designed for short reads could also be applied for long reads, or did you have to change a bit or twist it, the panel and all the conditions?
Yeah, so we actually explored this together with Twist. That's data I didn't show today, and we found that we needed a very different probe design to be able to capture the long reads. So we actually spaced the probes quite a bit further apart than what you would do with a short read approach, which is often more tiling based. Yeah, exactly. Yeah.
Thank you.
Yep. So if no one else have a question, I have one question, Suzanne. Do you need some improvement from the wet lab side, or is it really idle right now?
Yeah, where we've struggled a bit in the lab is with shearing. So that was quite laborious. We could use either the G tubes where you can spin 24 at a time, or Megaruptor where we could just do 8 samples at a time. But recently the field's brought a lot of new, more automation-friendly shearing workflows out there. So this is something that we're investigating, and then hopefully we can bring the plexity up, because at the moment we could run 24 samples on Sequel II and 72 on Revio, and that, yeah, requires quite a lot of sample prep in the lab. Yeah.
Sure.
Hello. I would just have a question. What about the other genes in the panel? Did you find something similar as a problem with CYP2D6 and copy number variations? Is this something missing? Have you found it, for example, in DPYD?
So I have DPYD in the panel, but I haven't reported variants from DPYD because we are still exploring this. So for those of you who aren't familiar with DPYD, this is a 1 megabase long gene. So it's actually half the content of our panel, and it requires a bit more of a closer look before we can draw any conclusions. But yeah, very good question. Yeah.
Okay. Thank you, Suzanne. Yep. Now I would like to welcome Professor Dr. Johannes Schocke from Institute of Human Genetics, Medical University in Innsbruck, Austria, and he is also talking about long-read panel sequencing, but with different project. So welcome, Professor.
Yes, thank you very much. Again, there are a few more seats here in the front if someone wants to sit down, but it's also nice to stand sometimes. Thank you very much to Twist for inviting me to give this presentation. My conflict of interest is really only that Twist will pay me for being here. And I would like to start with a disclaimer. I'm not a bioinformatician. I do not do the lab work myself. I'm a physician myself. And also, we are doing work in progress, so we are not where we want to be yet. So I show you results that I think are interesting, but we haven't actually started to really use the method that I present for diagnostic purposes, but I hope to do this in the next few months. So just to repeat what you all know, most of our work, so we do a lot of the diagnostic work for Austria, and we do most of it by short-read exome, and you all know that it's a cost-effective, fast, well-established method, very reliable, high quality, but there are important challenges. You cannot get the intron or promoter variants usually. We do not normally get structural variants unless they really interfere with the exons. We have a real problem with paralogous genes, highly repetitive regions, and also high GC content regions. So we all use it, and we all know that it's not good enough. So what are we all talking about? If you go around, we're all talking about genome-based diagnostics, which remains some of the problems, and then to resolve even further problems, and we had several meetings here about long-read genomes, which are lovely. They're great, you get methylation data, but they are very data intensive. They are even more expensive than short-read genomes, and they are not widely available. So this is not really suitable for the standard diagnostic purpose. We got a couple of dozen samples for cardiogenetics, for example, per month, so we cannot do thousands of long-read genomes in our diagnostic setting. It wouldn't pay in Austria. So that's why I really thought about a concept of doing long-read sequencing on a panel base, and that's the reason why I'm here. So a combination of the well-known from exome analysis, hybridization-based enrichment of all relevant genes for a specific indication. And it's important to clarify that for many of the indications that we have in daily practice, like kidney disease, heart disease, eye disease, we actually have a well-established set of panels of genes that we are interested in. Yes, there are undiscovered genes to be discovered, but it's unlike 8 years ago when we did panel short-read sequence where we missed all the new genes. We really know quite well for many indications what we are looking for. But we're looking for these in short reads and not in long reads. So and there are some problem genes that we do not cover with our standard method. So combine the well-known hybridization enrichment with multiplex, meaning cost-effective long-read sequencing. So what methods do we use? We use a twist, a custom-built targeted long-read gene enrichment, and that is very flexible. You pick your genes and they do it for you. We currently use for one run 8 samples because that's easy with the Megaraptor with a sample, but I think I will show you it's a very good coverage, so you could easily do more than 20, I would say. Per smart bell, so that gives you 12 samples for one smart cell on the review, which reduces costs dramatically. We use the PacBio SmartLink pipeline and use both IGV and the beta version of JSI for the data analysis. So this a view on the workflow. Can you actually see my? Yes. So, okay, you have the normal sample preparation, you add the DNA extraction. The hybridization takes about 3 days. So that's, overall, we take a week and a half for the whole process. It's not a rapid process. 3 days enrichment, then 2 days preparation for the long read on the review. Then we have the standard sequencing run on the PacBio review, and basically the initial data management is a day. So the total time for the workflow is a week and a half. The hands-on time for a dozen samples is about 12 hours, so it's about 1 hour technician's work per sample. So it's not automated yet. I think we will automate it, but it is reasonably time effective, I would say. There is a phase of a large number of bioinformatics tools which are collected here that we've used, provided by PacBio, and the visualization depends what you use, and I think there are some nice images that can be generated. So on our panel, we use 120 genes to start with something, and we combine it in a way that even if you only get 1 or 2 samples per week, we can combine it with other indications so that we can potentially run 1 panel every week or every 2 weeks, so make it cost effective. If you have a panel which you run only with 12 samples and you have only 1 sample per week, then you need to wait 3 months for having the number of samples available, which doesn't make sense. So we combine different diseases. We have the problem indication of cystic kidney disease, SMA, various repeat disorders. It's basically a kind of random collection of indications with the concept of allowing us to run one of those panels cost effectively per week. And it works very nicely for standard genes. So we have included phenylalanine hydroxylase. The gene causes PKU. Very simple gene, no paralogs, nothing. And you can see here, visualized with the JSI system, the 2 different haplotypes in the long read runs. And you can have 1 curl haplotype, and all these lines are individual variants. The gene goes down here from the beginning to the end. There is a gap in the twist probes, which is probably a lack of communication on our side, what exactly was our intention. So that will be solved in the next probe design. But even with this gap, we could have 2 contig haplotypes. So we can also throw this out and just look at the 2 haplotypes and then identify what variants are there, and are there any variants in cis or in trans? Is there anything in the introns? Use your standard diagnostic pipeline. So that's simple, and that goes for, I believe, for all the standard genes that we look at in exome analysis. So we have all the genes that you are interested and that are easy included in the panel, and we get long read results from before the promoter to after the 5' end. So what about the problem genes? One of the important problem genes is PKD1. There is the PKD1 gene, and there are 6 pseudogene homologs, which are really make it a problem if you do not find anything and you cannot confirm it. You're not 100% sure that you haven't missed anything. That's one thing. And the other thing which I would like to point out is that we have a conceptual problem. We think it's a dominant disease, autosomal dominant polycystic kidney disease, but you also have the possibility that you have biallelic hypomorphic missense variants, which in combination give you a PKD1 associated cystic kidney disease phenotype. So this is a nice review by Carsten Bergmann, where he shows, different constellations, and one of the constellations is, for example, 2 hypomorphic missense variants that on their own don't cause much problems, but in the kind of biallelic, recessive, but it's really, let's call it biallelic constellation, causes manifest cystic disease. And if we assume that we have missense variants that could cause this in a biallelic version, we have a problem, because if you go to GNOME ID and ClinVar and look at what kind of variants are in this huge gene, PKD1, you find there are 7,000 missense variants. 26 are pathogenic, but there are more than 6,000 that are unspecified. We just do not know whether in a biallelic version they could cause something or not. We may disregard it because we say, okay, we find this, and then we test the parents and they have it, so they have no cystic kidney disease, and then they say, okay, can't be anything because we think dominant, but we need to think biallelic. So what do we do when we actually find this, that we have two unknown missense variants, unclassified missense variants? Well, one of the things that we would like to know, is it in cis or in trans? If it's in cis on the same chromosomal strand, then we can look at it and say, okay, let's check the parents. We did this here in one sample with a patient with atypical polycystic kidney disease, two variants of unknown significance. One of them, rare. One of them was described in a doctoral thesis as a variant of unknown significance in an ADPKD patient. So it's all the problems that we have in daily practice. So the first thing that we'd like to know, is it in cis or in trans? And we can get that easily done with the long-read sequencing here with IGV visualized. There are 4,500 nucleotides apart, and you can nicely see in this not haplotype sorted early image that you have two different alleles. On one allele is the one variant, on the other allele is the other allele. And then we have a problem, because we do not know how to proceed. We cannot test the parents, because one of the parents will have the one, the other one has the other one. And whether they have cysts in the kidneys doesn't help us whether this is causative. So this is an open question. We can forget about the family studies, because they will not tell us anything except checking siblings who may be compound heterozygous as well. But it's the next step to really work on solving the problem of cystic kidney disease. What other problem genes are there? One of the, I put this in, although I'm not really happy enough with having done enough work with this. One of the repeat expansions that are notoriously problematic is the hexanucleotide repeat in C9orf72, which is here in an intron between alternative non-coding exons and is a major cause of amyotrophic lateral sclerosis and frontotemporal dementia. Normal repeat number is below 30, but it, as we heard yesterday, the repeat can be more than 10 kb. So that's a real challenge for everybody. It's very high GSC content. We can't read it in our hands. We can't see it well on the exome. With the panel amplified long-read sequencing, we have only, can show you only, we didn't do it systematically, but here you can see it's beautifully separated, two haplotypes, and there is a 4 repeat, so short, tiny repeat, nothing big, but I would say up to 30, I would really expect that we see everything, and then we have the haplotype information. So we can say this is homozygous, but we have two different haplotypes, so they are biallelic, so we can say it is normal. I do not believe that we would be able to detect large expansions, because it's PacBio is shorter than Oxford Nanopore, and we have shorter reads, but we can recognize the normals, and if it's abnormal, go on and say, let's go for some other method. Just what I failed to mention earlier is, what is the read length? Did I show that earlier? I showed it on the slide maybe. With the genome, PacBio genome in our hands, we are at something like 16, 17, 18 kb. The short read panel gives us reliably 6 to 7 kb. So it is shorter, but for our purposes, I think it is sufficient, and if you pay 5% of what we pay for a genome, it's worth thinking about it. So SMA is another of the problem genes. You have two paralogous genes. The important one is SMN1. If that's missing, you get SMA, and SM2 decides how severe it is. They are highly paralogous, and there's one variant in exon 7 that affects splicing and causes reduced transcription to 10% normal. So the high sequence homology precludes reliable short-read genotyping, and we can solve this, no problem, also with short-read panel sequencing, but I'm not happy enough with the results. So what do we see? Here, with the haplotype phasing, with a very clever haplotype phasing approach of PacBio, you have an easy feature of 3 different haplotypes. So there are 3 copies of SMN1 or SMN2, and you can see all 3 copies have the SMN2 specific variant. So we have 3 SMN2 copies and no SMN1. So it's a diagnostic finding. Very easy, but not good enough for me, for our general practice, because I don't know the complete biallelic constellation yet. The paraphase gives you basically hybridates all the reads to one gene, regardless whether it's SMN1 or SMN2, and then gives you the haplotypes. But you have 3 copies, and you'd like to know how are they organized on your chromosome. If you have 2 SMN1 copies and 2 SMN2 copies, you would think, okay, one is on each chromosomal strand is one SMN1 and one SMN2. But we also see in a few individuals that there are 2 SMN1s on one chromosomal strand and 2 SMN2s on the other. So that person is a carrier. And when we do predictive testing, a partner of a carrier wants to know what's the risk for SMA. We say, yeah, probably you're okay, because it looks okay, but we cannot exclude that you're a carrier for an unusual constellation. And I haven't seen a solution for that yet. So we should be able to make a contact of the 2 chromosomal strands that allow you to say, okay, this is strand 1 from the mother. I could test the parents, obviously, but we do not want to test the parents. That's why we're doing this. And one is from the father, and we have it solved. So I think we will manage. If you look at this here, these are the 3 haplotypes. If you look at the data, with the IGV data, here you have a large number of unassigned sequences. So there are sequences that link to the locus, but you cannot link them to either of the 3 haplotypes. So I think if you be able to really group this together and see this must be the connection from the one haplotype to the next, we need to get a method in which we can kind of make a contact of 2 and 1, SMN1 or 2, here it's SMN2 copies, and then we are flying. Then we don't need parents, no family results. This similar problem is also true for, and then it becomes even more complicated or easier also in a way, with congenital adrenal hyperplasia, which is a very complicated locus. Here, the chromosome, the 6 short arm of the chromosome 6 locus, several interesting genes, tenascin X, here TNXB gene, the SUB21A2 gene and the pseudogene, and you have duplication variants where you have 3 of these gene groups, and you have various hybrid alleles. You may have a hybrid allele between SUB21 pseudogene and gene, or you have the hybrid allele between the tenascin X genes, which are also interesting for us because they are important for hypermobility and the CAHX disease. And that's very challenging. When we used our sample, we have a duplication. We tried out with the duplication sample. It is easy to sort the 5 different haplotypes, which you can see here. There are 2 sub-21 pseudogene haplotypes and 3 sub-21 gene haplotypes. And you have the whole sequence, and in 2 out of the 3 sub-21A2 genes, so the functional genes, you have the stop mutation here, which we know from the duplication. We know that there is one duplication allele where one of the copies has a mutation. But 25 years ago, we regarded this as a carrier status, and then I remember writing correction reports because we realized, oh yeah, there are 2 copies of the normal gene. Here it's a carrier because the other one obviously has the pathogenic variant as well, but really we would like to see the biallelic constellation. It has to be seen whether 6,000-7,000 nucleotides is sufficient for that, but I think we will get some answers. So where are we not really where we want to go? The kind of, in my opinion, the holy grail of long-read sequencing is lipoprotein A, the LPA gene, which has these Kringle repeats, which are essential for the LPA protein. So there are 10 Kringle 4 repeats, and the second one is available in 10 to 50 copies. The more copies, the better, because you have lower LPA concentrations. And high LPA concentrations is one of the #1 killers in Europe. High lipoprotein A, everyone in the room who doesn't know the lipoprotein A concentration, please get it tested, because if it's high, you may have a high risk for heart attacks and cardiovascular disease. And you can test it, you can measure it, but the genetic characterization is notoriously difficult because you have these huge number of copies, and each copy is 4 to 5 kb. So that's a real challenge. There are also various functional variants in these Kringle repeats which impact your effect. You may have a stop mutation which has an effect which you cannot really see, so it's very difficult to characterize. When we do that it's also on the dark gene panel of Twist, but I'm not sure that anyone has been able to actually get the result they wish. We haven't. In the panel enrichment, it works very nicely for all the Kringles except the Kringle 242 region, which you're happy to have 20 repeats on one and 30 on the other. But if you see the coverage here, there's huge kind of mountain peaks. It's like Innsbruck, where you have a lot of reads which are not assigned to anything. There are 6 Kringle repeats in the standard sequence here, but the gray things are all reads that we cannot link to a haplotype and link to basically a contig. Everything else, blue and orange, are the 2 haplotypes, so that's easy. But we haven't solved the Kringle 4-2 region, which is really why we're doing this. So I think let's hope that in the next 12 months we'll get this done somehow. It's a question whether the 6 to 7 kb coverage or size of the panel long reads work. It's even a question whether the 15 to 20 kb work. I asked yesterday the question whether Oxford Nanopore can read the Kringle repeats. I'm not sure that they can. So that's a holy grail of genetic diagnostics, I would say. So that's all I want to present. It's work in progress, so we haven't really done large studies with this. The concept is to enrich target genes that we find relevant, and not just our problem genes, but use this as a first-line tool, because with the PacBio results, it's really high quality, and you know that the data are reliable. So you include all the genes that you're interested. You get the introns and the promoter regions for the standard genes, plus hopefully all the problem genes. You get all the structural and sequence variants. Let's talk about the price. If you can do a dozen samples on a PacBio a smart cell, you are in the range of 100 euros per sample. And then the twist costs also something. But you are at a lower cost than at an exome for long reads that answer our question. So I think it's a cost-effective way if you can afford the Revio, which is an expensive instrument. Cannot extend this to other genes that you might be interested. Obviously, that's a nice thing about exomes, that you can indication changes and yet look at other genes. But I would say we know for the majority of the requests that we get for routine diagnostics, we know what we are looking for, and we never use the 20,000 genes that we could look at, and we look at our cardio or nephro or eye genes only. The read length is about 6 to 7. In our hands, the genome is 14 to 17, so it's not that big, I would say. Read quality is excellent, and we have some information that some repeat sequences get lost during enrichment, and it's work in progress. Challenges are regions with high GC content. We haven't solved MUC1 yet, and we need that for the kidney disease. Also, AG content is relevant. How to visualize the repetitive sequences as a context is a challenge to the people who provide the software. So the allelic constellation is something we want to have, and we need to explain why we want to have it. Probe gaps can be solved, and there are solutions for all these challenges, I believe. So with that, I thank you for your attention and happy to answer questions.
Thank you, Professor, for the nice talk. So stage is open for question and answers. If you have any questions for Professor, please, happy to ask. So Tina has.
Yeah, can I answer your question? Because you actually discussed the LPA gene, right? Mm-hmm. The LPA gene is in the twist dark genes panel. We actually just have a preprint out on the coverage of this gene. So we should, so they were able, the Baylor, our collaborator at Baylor, were able to resolve it very well. So I think we should talk about it because it's actually working in the dark genes panel. So maybe.
I haven't seen the data that you can separate the kringle for 2 repeats.
Yeah, so I think it could be something about bioinformatics pipeline because, you know, Baylor are very skilled at how to, you know, do structure variation calling properly. So I think we should have an offline
Let's talk about it. And we present the data. I'm very happy if we can solve this problem.
Yeah, thank you.
Thanks for an awesome talk with lots of discussions of haplotypes. But I have a different kind of question. What are you using as a LIMS system for all of this?
For the bioinformatics, or for LIMS, I mean the...
for LIMS, yeah, to store all the phenotype data and so on and so forth.
I mean, we use Gepardo, if that's it's a Gepardo as a laboratory system, and we do different, basically a collection of different software for different purposes. We have for the variant visualization, IGV and JSI, which are nice. But I think any LIMS would work. I think there's nothing special about there's no specific requirement for the LIMS, I would say.
Okay, so it's working all right with the Revio data?
With most of the data that were analyzed, the main bulk of the analysis was with the PacBio software portfolio.
Thanks.
Thank you all for attending the Corporate Satellite Symposium.